panelists, our participants on Facebook and Zoom a few more seconds to join the 11th session of the Bahamas National History Conference, and then we'll get started. My name is Pachancia Knowles, and I am a Marine Science Officer at the Bahamas National Trust, and I will be your moderator for today. Good morning once again, and welcome to the Bahamas National History Conference, Session 11, Into the Deep, Exploring the Depths of the Bahamas. And this session is brought to you by the Bahamas National Trust, and we will have all of our panelists present first, and then we will hold a question and answering session at the end. So feel free to provide your questions in the Facebook comments or if you're on Zoom, in the question and answering section, and we will answer all questions and direct them to the panelists at the end of the session. And this morning we will get started with Dr. Charlotte Dunn, who is one of the directors of the Bahamas Marine Mammal Research Organization, and she will be speaking on listening in the deep. Dr. Charlotte.
Thank you, and nice to hear your voice again. Pachancia was one of our many interns over the years. So I hope everyone is staying safe and healthy, and I can provide a distraction for a few minutes. I am on the left here. I'm Charlotte Dunn. I'm Bahamian. I'm an acoustician, so I study sound. My research is largely aimed at understanding the impacts that sound makes on deep diving whales in the Bahamas. I am a director of BIMRO with Diane Claridge. We are based in Sandy Point in Abaco. We're both research fellows at the University of St. Andrews in Scotland, where we did our PhDs, and we are adjunct professors at the University of Miami. So I'm pretty excited that the BNT have focused an entire session on the deep. It's often forgotten. Our banks are so beautiful. We get photos sent to us from astronauts, and you can actually, however, see that between our islands, we have quite a lot of deep water. Some of the water, like between Abaco and Nassau, goes to 2.5 miles deep. But the Bahamas is largely what we call an oligotrophic environment. So that means that there's actually limited nutrients. And some people describe the banks of the Bahamas as having gin clear water. Oh, don't say gin, our liquor stores are still closed. Anyway, the reason it's so clear is because there's nothing in it. But in some of the deep water where the marine mammals feed, it is actually quite productive. And these, deep diving whales, there's a sperm whale in the top left and the bottom right is a beaked whale. They're feeding down at 1,000 meters, so about 3,000 feet. And there they are finding the prey that they need. And that's why the deep water of the Bahamas is very important for marine mammals. And as the other speakers will tell you, other species as well. So I want to talk to you largely about sound. That's my specialty. I think some of the older Bahamians may recognize this song. It's probably the most famous Bahamian song ever recorded by Ronnie Butler. And maybe you remember the last time you danced to that song, or maybe you can remember the last time you danced at all. I remember as a kid, my dad putting me on top of his shoes and trying to teach me rhythm to that song. But sound for us is all around us, and it's constant. Right now, I think, unfortunately, friends in Abaco are probably still hearing generators. Other people maybe are hearing birds. As you know, the world has become a little bit quieter. We can hear nature a little easier. But I want you to think about what's happening under the surface of the ocean. Once a whale gets beyond 200 meters, it's completely dark. So that whale uses sound for everything. They use sound to figure out where they are, communicate with each other. They use it to find food, and they also listen. So they're listening for a threat like an oncoming vessel or possibly a predator. This is an adult female beaked whale in South Abaco, and behind her is an adult male. You can see the sunlight coming through. These animals are completely silent at the surface, and as soon as they hit 200 meters, we know from tagging them, they start to use sound to, for all their life functions. Check this guy out, brand new sperm whale baby. This is last summer in South Abaco. If you listen, you can hear clicking, and that clicking is this animal's mother feeding at depth about 1,000 meters deep. So she's left her baby with this babysitter, and they are actively following those clicks. So when the mother surfaces, they'll be nearby. The mother is making those clicks, and the sound that echoes back from those clicks will tell her where there's squid and fish that she's looking for to feed. But we also do have predators here. Some killer whales in August last year in Andros came up to the boat, and they will be listening for other marine mammals' sounds. We've seen killer whales feed on marine mammals in the Bahamas over the years, and the marine mammals, the other marine mammals, will be listening for the killer whale sounds as well. So sound it's everything for deep diving whales. What about the sound that we put in the ocean? So we know since the beginning of time that cargo has been transported on water, whether it was attached to logs, and then in the 1800s, steamboats were invented. So steam was used to turn paddles or turn a screw propeller. And then in the 1920s, mechanical screw propellers were invented. And that was really the introduction of noise pollution into the ocean. This is NASA in the 1900s. And here it is last year. So this is the noise that the whales hear when ships go by. It's throughout every ocean, and it is the key factor of noise pollution in the water. 50 years ago, a blue whale could communicate with another blue whale from one side of the Atlantic to the other. But in the last 50 years, the distance they can communicate has decreased by 90%. This is the port in Freeport. It's expanding. We know that ship noise could double by 2030. This is a real threat for the Bahamas. The Northwest Providence Channel is an international shipping lane. But we also know if ships reduce their speed by 10%, we can reduce underwater noise by 40%. And if we put into regulation some of the voluntary guidelines that the International Maritime Organization have put forward, like standardizing ship noise measurement protocols for ports in the same way it's done now for airports, we could really address some of these issues. Some other sound that is put into the water and also in the Bahamas, these are air guns, that sound. In the same way the sperm whale puts out sound and the echoes tell it where its prey is, air guns are used to find oil. So that sound is going straight down, and the echo that comes back from the seafloor informs whether there's oil in the seabed. This is the loudest man-made sound in the ocean, and they travel about 1,000 kilometers. In 2018, a spotted dolphin stranded in the Berry Islands, Atlantis in Nassau, and released it in Bimini, where on release, it swam for 48 hours over 400 kilometers, and it hung out in this area, and that circle is surrounding the Bahamas Petroleum Company's drilling site. They haven't started yet because of COVID, but there are no regulations. They've already conducted the those air guns, the exploration, and there's no regulations for the drilling either. This story is in press in Aquatic Mammals that will be out shortly. But we saw last week in the devastation occur in a small island nation when there is oil drilling or exploration. Some other sound that we have here is the sonar testing that's done at the Navy base in Andros. It's the same method. The ship puts out the sound, and if the sound hits a submarine, the echo goes back to the ship, telling it where, if there's a sub there, which direction it's going, how fast it's going, and where it is. So there are small resident populations of beaked whales in the Bahamas, and they are particularly susceptible to sonar, and they also have high sight fidelity, so they don't move around much. We have put acoustic tags on these animals, and then we can see that when they're clicking, which is the sound they're trying to find food with, and sonar is played, they stop clicking. This animal eventually stopped clicking altogether during the sonar test, and you know, this has effects on that individual's fitness and the population. Something else to remember is that all the fishing boats, visitors and local, are now using echo sounders more and more to find fish, and also to make sure they're in deep enough water. They put the sound out and the echo informs them. We now know that echo sounders are also affecting marine mammal foraging. So the effects of sound in the water on marine mammals can be immediate, live strandings, getting away from the sound, or long-term. So they can, you know, it can affect their hearing. Well, if you think of all the things we've talked about and listening for echoes, if your hearing's impaired, that's pretty detrimental. Changes in vocalizations, many species, terrestrial as well, are having to change their vocalizations. We know killer whales are increasing the loudness and duration of their calls. Behavior, we know that the animals at the Ortec range are leaving and returning after tests, and this is affecting their reproductive success. And we also know it's affecting animals' physiological stress levels. So if we're in a loud, open plan office, we can just put on a headset, or we can work from home that day, or now work from home that whole year. But animals don't have those choices. So it's really important to consider that. So just a little bit, some of the research we're doing at the moment, we fly small hexacopter over beaked whales in Andros and Abaco. This is an adult male and female and a calf between them. And then we take high quality photographs. We can measure the length and also the girth. We can infer pregnancy and get growth rates, which helps to monitor the population status. Another project we're doing is the shared waters. So we're looking at how ship noise and ship traffic and sperm whales have overlapping habitat. In the bottom left, there's a ship traffic heat map, and it shows the traffic coming from the Atlantic going to Freeport or to Florida. And in the top right is this sperm whale use map in pink from sperm whales we've tagged. And in the top left is one of the recorders that we use part of this project. So this recorder, actually, we put a hole in the wall, and we are collaborating with the Perry Institute of Marine Science. They had anecdotal information of a grouper spawning aggregation there. And I highly recommend collaborating when you have really expensive equipment. So this recorder will tell us what was the noise of the vessels that we can see on the AIS, the Automatic Information System. How many marine mammals were there? We'll be able to detect marine mammals. And also now we'll also be able to tell us about groupers. So that's a great collaborative effort. We will, from this information, hopefully be able to inform government. Do we need a slowdown zone? Do we need a shipping lane? And some of this data now is being contributed to the International Quiet Oceans Experiment. So what the scientists are now referring to COVID as an anthropause. These ships, these photos, one is Philippines, one's from Europe. There's ships drifting all over the world. We've seen it in the Bahamas, around the Berry Islands. In Northwest Providence Channel on the AIS, you can see tens of ships just drifting. Well, we know that with ships reducing their speed by 10%, we can reduce underwater noise by 40%. So the whales are getting some respite. I know these ships are probably disposing of their rubbish, and there's light pollution, but we also know that all of this quietness is giving the marine mammals a chance. The Vancouver port has reported 15 dB less power when after 2011 with 6 dB less power, whales showed a significant difference in their stress levels. So for some of the animals, they're having less stressful time than we are right now. But let's learn from this and try not to go backwards. And I just want everyone to think going forward about sound for marine mammals as well as just ourselves. And hopefully we can all dance to this song together sometime soon. Thank you.
Thank you, Dr. Charlotte, for giving us that insight on man-made noise pollution and how it affects our marine mammals and all marine life. We will now have Dr. Stephen Kessel from the Shed Aquarium to now share with us on his studies.
Thank you very much. Thank you all for joining this session. Today I'm going to talk to you about it's a preliminary kind of proof of concept survey conducted last year in the tongue of the ocean around New Providence. I'd first like to acknowledge my co-authors on this talk, Dr. Lester Giddens, Brendan Tawa from the Cape Luther Institute, Brian Moe, and Dean Grubbs from Florida State University. This survey was designed to survey throughout the deep sea ecosystem, but its initial concept, I'm just trying to figure out how to get my slides to advance it. You're going to work for me? Technical difficulties. There we go. So initial concept was based on sharks and rays. This is part of the Shed Aquarium Shark and Ray Research Program. So I'd like to start by quoting this paper by Dr. Grubbs and Chip Cotton from Florida State University that states approximately half of all known chondrichthyan fishes, that being sharks, skates, rays, and chimeras, live in the deep ocean, and the deep ocean is classified as anything below 200 meters. Yet only a very small proportion of current peer-reviewed publications focused on elasmobranchs look at deep sea species, only around 7%. And there's a fair few shark and ray publications that have come out of the Bahamas, but I'd say the vast majority, less than 7% in the Bahamas, are focused on deep species, the most being focused on shallow species. And we know there's been a lot of research over the years from groups like the Bimini Biological Field Station and Cape Eleuthera Institute and others on the shallow species, but definitely far less on the deep species. And we talked about the shallow banks of the Bahamas and how they're intersected by these very, very deep ecosystems that are relatively largely unstudied. This highlights 3 of these, being the east side of the Gulf Stream off of Bimini, the tongue of the ocean between Andros and New Providence, and the EXUMA Sound. And I've highlighted these 3 areas as these are 3 areas that have received a little bit of research in the deep environments from a shark and ray perspective. The Bimini Biological Field Station has been conducting surveys off of Bimini for about 20 years now, not so consistent, but increasing in that consistency. Cape Eleuthera Institute has initiated a survey of the EXUMA Sound. And I know that the Cape Eleuthera Institute, with members from Beneath the Waves, have done some research of the deep ocean and deep sea sharks in the tongue of the ocean. And more recently, in collaboration with the Cape Eleuthera Institute and others, we have initiated this survey, but still in its very early stages. My motivation for the survey came from some of that work back in the early 2000s in Bimini. I was part of those experimental deep long lines, and we pulled up some very interesting deep sea creatures, including this guy here, which at the time was known as a big eye 6-gill shark, has since been reclassified as an Atlantic 6-gill shark. But much to my delight, coming to Shed and working back in the Bahamas and speaking to Dr. Lester Gittens from the Department of Marine Resources, he expressed the desire to have a better understanding of their deep sea ecosystems, including the tongue of the ocean. The research program from Shed Aquarium, we are based here in Chicago, where I currently sit in my apartment, kind of trapped by the pandemic. But what I should be doing is being down in the Bahamas on our research vessel, the RV Coral Reef 2. This is our slightly larger than 80-foot research vessel. It was originally built as a sustainable collection vessel back in 1984 and has since been partially repurposed as a research vessel. When I came to the Shed Aquarium, having an interest in starting to survey some of these deep sea environments, one of my first questions to my boss, Dr. Chuck Knapp, was, does this boat have a winch? And much to my dismay, the answer was no. So that was a little problematic. As working in Bimini without a hydraulic winch, I really understood the need for a hydraulic winch to really make a dent in some of this deep sea research. So this very early survey, including the one this picture was taken from, we pulled these lines up by hand from around 800 meters. And that was not ideal or efficient by any means. Again, I've got to really credit my boss, Dr. Chuck Knapp, because together we petitioned the Shed Aquarium to purchase a winch for the boat to allow these surveys to be conducted. And it took a lot of work on his part to convince them that spending around $50,000 to make this happen was a good idea. And amazingly, he was able to do that, and I'm very appreciative for that. And based on a recommendation from Dr. Grubbs from Florida State University, this is the same winch he uses for his deep sea survey of the Gulf of Mexico, upon which the survey is largely based. This is a Lingua-Pittman hydraulic winch and spool, and it's powered by a hydraulic power pack, which taps into the hydraulic steering fluid for the steering system for the boat and runs off a power pack bolted to the port engine. Then in order to properly operate the winch safely, we had to purpose build this A-frame. And this really came down to the wire. This was back in last September when the first trip was initiated. This was the morning of the departure for the Bahamas, and you can see the guys still helping to bolt and weld the A-frame and winch onto the boat. So I was a very happy person indeed when we were able to finally get this winch turning and spooling on that line. Here it is fully spooled. We put 12 kilometers of twisted nylon tarred line again, the same they use in the Gulf Deep Sea Survey, Gulf of Mexico Deep Sea Survey. The reason so much to 12 kilometers is this way we're able to conduct a series of sets simultaneously and maximize the survey potential for the region. Here on the right, you can see a little better the purpose-built A-frame at the very top there. There is a block that the line runs through and keeps the line away from the boat and the engines when we're hauling and setting off the rear of the boat. So some of the survey details, again, it's a preliminary survey, so I don't have a ton of data, unfortunately, to present today. This second survey was supposed to be conducted next month, and unfortunately, due to the pandemic, I don't think that's going to happen this year, but this is intended to continue into the future. So to date, we have only conducted one survey in the region that ran from the 7th to the 14th of September 2019. The survey was developed in partnership with Dr. Lester Giddens of the DMR, and the methods were based off of the Florida State Gulf of Mexico survey in like close consultation with Dr. Grubbs, who is one of the leads for that survey. We had an additional consultation from Cape of Good Institute, Brendan Talwar, in particular pictured on the left here, and he talked to us about and helped us with some of the nuances of doing deep surveys in what he had found in the EXUMA Sound that helped us have success in the tongue of the ocean. This was also a project in partnership with the Association of Zoo and Aquarium's Safe Shark and Ray program. SAFE stands for Save Animals from Extinction. And through that, we were able to partner with the Georgia Aquarium and Audubon Aquarium in New Orleans. And we have participants from both of those organizations, that being Dr. Lisa Hoops on the left and Ashley Newmeyer from the Audubon Aquarium on the right. Here's the setup of the line. This schematic was adapted from one said to me by Dr. Grubbs that was constructed for their deep sea program in the Gulf of Mexico. Just going to go through the setup of each of the deep lines. So the setup is essentially the same. The only thing that really varies is the scope of the line that goes to the surface, and that's essentially 1.5 times whatever the intended depth is. The depths we were setting at was aiming to be between 200 and 1,000 meters. The line from the anchor upwards, it starts with about a 5-pound grapnel anchor to sink it to the bottom and keep it there. We then have a fish trap with 1 kilogram of Manhattan chum. We have a series of traps and hooks on the line, and that's intended to sample both the fish, elasmobranchs and teleost, and any invertebrates that may be down there. Since we're going to all the effort putting this gear down, it's best that we can survey all the elements, or as many elements of the ecosystem as possible. We then have a series of 30 gangins that are intended to stay very close to the bottom. A mixture size of circle hooks from large 18-aught at the upper size, intended for the larger elasmobranch species, all the way down to 10-aught circle hooks for the smaller teleost and elasmobranch species. All of these gangins were baited with bonito. We then have a smaller shrimp trap for some of the smaller invertebrates at the end of the line with again 1 kilogram of Manhattan chum, about 5-pound lead sash weight just to keep the line and make sure the hooks are kind of staying near the bottom, and a temperature depth recorder so we can see exactly how deep the line was set. We aim for these different depth ranges, but as the line sinks and drifts as it falls to the bottom, it takes it quite a long time to get to the bottom. They don't always end up exactly where we intended, and we really need to know what the depth we were surveying at was for each line. The lines are deployed off the stern of the boat. We keep the boat in gear traveling forward, and the line free spools off the back. We have 2 people positioned on the back of the boat and a series of people handing them hooks and traps and gears over the transom. Meters with 4, 5, and 6 hour soak times. The logic behind these soak times is we're trying to have the gear surveying on the bottom of the ocean for 3 hours, and it takes a long time for that gear to get down there, especially at the the deeper depths. So these soak times are designed to allow the gear to go down and settle for around a 3-hour period. The line is then hauled again from the stern. We coordinate this with the captain who can't see what's going on via VHF radio. And all the specimens, as they are brought to the surface, are either removed from the line and passed to the back of the boat where they're transferred to the live well systems or coolers full of water. Or if they're larger elasmobranch species such as tiger sharks or similar, we work these up along the dive platform. We're able to do that safely because of the setup here. So here is our survey results. We were able over that period to conduct 10 surveys in total, varying in depths from the shallowest being 238 meters to the deepest at 1,085 meters. I would have liked to have conducted more surveys. We were aiming for 3 sets per day, basically one aimed at 200, one aimed at 600, and one aimed at 1,000 meters. We had varying issues along the way, and this being a preliminary survey, we had to work out the kinks. We had some mechanical issues where the hydraulic container was emptied by an unconnected overflow valve. That also meant we didn't have any steering on the boat for a period of time, and that set us back by about a day. We were dealing consistently with weather issues, and that's why the survey is distributed as it was. Initially, we were able to survey to the northeast of the island where those first 4 sets were conducted. After that, we had to switch based on a change of winds and to find lee off of the southwest part, kind of over near Stewart's Cove there. But we did get 10 sets in. We were also chased out, and we had to lose a couple of days of the trip because there was a pending hurricane moving in to the New Providence area, and we had to run from that back to the US. So we lost a couple of days, unfortunately. And originally, these were tended to be very good weather days, so I was quite disappointed there. Just to give you some preliminary results, this was just a pilot study, but we do believe it was successful. We caught a total of 6 sharks of 6 different species, a total of 7 individual teleos from 5 different species, and a total of 49 invertebrates from at least 7 different species. I've also broken these up by different depth ranges. So this is everything we caught between 200 and 400 meters. We caught a couple of elasmobranchs in a dusky smoothhound and a tiger shark. We caught 2 fish species, an almaco jack and a mutton snapper, and a couple of invertebrates, both being arrow crabs. Moving up between 400 and 700 meters, we pulled these sets. We caught again another couple of sharks. We got another Atlantic 6-gill, like we had previously caught in Bimini, Cuban dogfish. This is the depth range, and these were all on the first line we did at 405 meters. This is where we caught all the kind of deeper species of snapper, a total of 4. Not a huge amount, but we did get some. We also caught some giant isopods, which are very cool animals to see. We originally thought these were all Bathanomus giganteus. And since then, with assistance from one of Brendan's contacts at the University of Florida, Gustav Paulay, who is an invertebrate expert, we have confirmed that some of these are the species Bathanomus maxiorum, which were previously described only a few years ago by CEI and their team off in the EXUMA Sound. And what's quite exciting is this is the first place outside of the EXUMA Sound that the species has been sampled. So the tongue of the ocean EXUMA Sound are the only known range for the species to date. We also caught a brittle star down on some of these deeper lines nearer to 657 meters, a lobster species, and a shrimp. Finally, at the kind of deeper end of the spectrum, we did pretty well on the invertebrates here. There's a lot of these giant isopods lurking down in the depths there. We had a teleos species being an oilfish, another species we previously sampled in Bimini, and thankfully Dr. Grubbs, when we caught some Bimini, informed us that the scales on these oilfish are incredibly sharp, so I was able to avoid lacerating my hands this time because I knew what I was dealing with. Again, a lot more of the giant isopods. 2 deeper species of shark, the common gulper shark, and also the rough-skinned dogfish, which were very, very cool to see, and another species of deep sea shrimp along with an amphipod. So just to summarize, as I say, not a ton of data. This is our first survey. This was a proof of concept trip, but I believe it was successful, and we have every intention of going forward with this. We, as I said, we're scheduled to go out next month for this, but we're going to have to push that back to next year as soon as we're able. I was a little disappointed with the total number of sets we're able to conduct, and that was impacted by mechanical and weather issues. We've since resolved these any potential for those same mechanical issues by putting the hydraulic system for the winch on a standalone system to not compete with the hydraulic for the steering system. Overall, I'd say from what I was hoping for, maybe we had a relatively low catch per unit effort, but definitely some very interesting specimens coming up from the deeps around New Providence. We had a pretty similar ratio in terms of the number of sharks to teleosts, but overall the biomass, because of some of those larger sharks, was greater for sharks and teleosts. In going forward, we do plan to continue the survey, and we would like to increase our level of coordination with the Cape Luther Institute survey and the Bimini Biological Field Station survey. Maybe also coordinate use of the vessel in Bimini to use this hydraulic winch and increase the potential to do these surveys in the kind of eastern side of the Gulf Stream, where I know there's a lot of other interesting species. And we've also been in discussions with Beneath the Waves, Austin Gallagher and co Beneath the Waves, of the potential of tracking some of these deep water shark species around New Providence. There is an installation of deep sea receivers, as well as some of those shallow acoustic receivers. So we'd like to implant some of these deep sharks with acoustic transmitters and see what kind of spatial ecology they have around the island. Just like to quickly thank the crew of the Coral Reef 2 who make all this research possible, all the different participants. We had a good range of people from Cape Luther, Georgia Aquarium, Audubon Aquarium, Florida State University, Brian Moe, Hannah Rutson from the American Shark Conservancy, also Walcott Miller, who I'm sure some of you on this call know, always a pleasure to have on the boat. And finally, some funding from the Shed Aquarium, Georgia Aquarium, Audubon Aquarium, and Paul M. Angle Foundation. Thank you.
Thank you, Dr. Castle, for sharing your data on the Tongue of the Ocean deep sea surveys. That was quite a number of species, and it's interesting to know what is being found at each step. I want to turn our attention to the Natural History of the Bahamas book. The National History of the Bahamas is a field guide that is small enough to carry on trips, yet is an ultra comprehensive guide of the terrestrial and coastal flora and fauna, including species and common names, locations, and detailed descriptions. The book was written by experts in the Bahamian ecology, with contributions by more than 30 other naturalists and researchers, and covers organisms studied in the Bahamas and the Turks and Caicos Islands with more than 700 colored photos. It is available for sale in New Providence at Logos Bookstore and Media Enterprises, as well as all national park offices that the BNT manages throughout the Bahamas. The book can also be purchased online at Cornell University Press and online book retailers, such as Amazon for Kindle instantly and by mail. All royalties from book sales benefit the Bahamas National Trust. So it is a great way to support the Bahamas National History Conference. So moving on to our third panelist, I will invite Brendan Talwar from the Cape Luther Institute to share his screen. And I just want to remind everyone that if you have questions for any of the panelists, please provide your questions in the question and answer box below, as well as on Facebook if you're viewing from Facebook. If you can provide your questions on the comment sections, and we will answer them at the end of the session. Brendan Talwar.
Yeah. Hello. Can you hear me?
Yes.
Perfect. Well, thanks everybody. Thanks for being here. My name is Brendan Talwar. I work at the Cape Luther Institute. I'm also a PhD student at Florida International University in the US. Today I'm going to share a bit of work that we've been doing on the offshore habitats here in the eastern Bahamas under an umbrella called the EXUMA Sound Ecosystem Research Project. So to begin, what you can see here is a view along the wall at about 60 meters down. To the left is a vertical cliff face stretching from relatively well-studied shallow habitats down to the ocean floor, hundreds of meters below, and the pelagic zone to the right, both of which we know very little about. And the open ocean in much of the world, including the subtropical Western Atlantic and the Caribbean, is often referred to as a vast desert. We think of it as a place that lacks any kind of structure and is generally devoid of life, and sometimes that might be the case. Much of the time, there's a lot more going on than meets the eye. In reality, these habitats hold significant populations of fast-moving, large, energy-demanding predators that are often the target of commercial and recreational fisheries, and most of the time their distribution is quite patchy. But oceanographic features do structure this environment. Things like upwelling driven by seamounts and steep walls, tidal movement of coastal nutrients into offshore waters, currents, eddies, fronts. There are lots of features out there that we just can't see with the naked eye. So here you can see a map of the eastern US and western Bahamas on the left, and a zoomed in image of Northwest Providence Channel on the right. The colors represent modeled vorticity and energy dissipation caused by currents as they flow along bathymetric features of Northwest Providence Channel. This is from Gula et al. 2016. And it's really cool. I just found this paper a few days ago, actually, but you can see there's this semi-permanent cyclonic eddy that rests between the Little Bahama Bank and the Great Bahama Bank. And that's exactly the type of oceanographic feature that would attract pelagic predators. So features like this are structuring the pelagic environment. They create hotspots of biodiversity and abundance that are often critical foraging grounds and maybe more for large roaming predators that sometimes spend a lot of time in Bahamian waters. One example being the oceanic whitetip that aggregates just south of Cat Island. And if you go deep enough, you can find immense biomass held in the mesopelagic zone and incredible diversity along the seafloor, as Steve was touching on just now. And so there's this paradox between what we think of these habitats and imagine them to be and what they actually are and the life that they hold. Here in the Bahamas, these habitats are immense. They cover the vast majority of the Bahamian EEZ. Right here, we're looking at the central Bahamas from the north. You can see EXUMA Sound, Tongue of the Ocean, and all these other deep open ocean habitats, some of which are very unique. The deep water basins like these are really not common. The large amount of edge habitat that forms their shallow boundaries means that there's a constant interaction between deep water, open ocean habitats, and those shallow reefs and mangrove and seagrass communities that we know quite well. We've even heard stories from fishermen from time to time of catching mahi and tuna on the fly in like 3 feet of water up on the banks. So that to me really demonstrates the connectivity of these ecosystems. They're tied together far more than we realize, and that connectedness might give them a bit more resilience in the face of climate change or other anthropogenic stressors. So it's something that we definitely need to learn more about moving forward. But because we know so little about them, we have a hard time managing them or conserving the habitats or the species that live there. It's quite difficult. So we definitely need to know more about life in the open ocean and fill in that piece of the puzzle in our understanding of tropical marine ecosystems. And this paradox of thinking about these habitats as empty and lifeless when in reality they host incredible assemblages of marine life is really just due to a lack of understanding. So what we need to do is get out there and get down there and take a closer look. So to fill in some of these gaps, my colleague Eric Schneider and I started the EXUMA Sound Ecosystem Research Project. It's just an umbrella initiative that we've been working under, and it's what I'm going to focus on today. So our goals are quite broad. First, we seek to educate and engage students. We work with kids and students all the time. Basically, an educational focus is blended into everything that we do. If we're doing some kind of research, we're actively teaching from start to finish. We also seek to understand the dynamics of a unique subtropical, oligotrophic, or nutrient-poor ecosystem like EXUMA Sound. You know, how does that system change over time, seasonally, or over a few years, or over many years with climate change? How does it change with the introduction of fish aggregation devices, or if management practices vary? You know, for instance, if size regulations go in place for a species of pelagic fish, how might that impact the wider food web? Or if coastal development occurs near a biological hotspot, how might it affect the food web of that area? So through studying the system and modeling it, we can begin to have a shot at predicting some of those outcomes, ultimately informing marine resource management, particularly related to siting marine protected areas. So to get to that stage, we have a lot of basic research to do. Our research themes are quite broad. We hope to characterize abundance and diversity of a wide range of organisms in the pelagic zone. from invertebrates to highly mobile marine megafauna, and then understand habitat connectivity and complexity from the mangrove ecosystems out to blue water, which in a place like Eleuthera, you know, those systems are just a few kilometers apart. And then also understand predator ecology, specifically movements and trophic ecology, and how these animals might act as vectors for nutrient transfer between these different habitats. And then, of course, how all of these things relate to anthropogenic impacts with a focus on fisheries in our case. So EXUMA Sound is about 10,000 square kilometers. It's a semi-enclosed basin. There are only two openings, one at the bridge just south of Eleuthera, in between Eleuthera and Little San Salvador, and another south of Cat Island. And there are diverse habitat types here in close proximity to one another, and it gets very, very deep, over 1,600 meters in the middle. So one of the first things that we needed to do was map the seafloor. So the team at CEI, a big group of people, worked with OceanX and then Biolucia to get this done. We now know what the bathymetry looks like just off of Eleuthera. So if you're not familiar with this island, what we're looking at is Cape Eleuthera highlighted there, and then that's the whole kind of southern extent of the island. And to the southwest of that is EXUMA Sound. So now we can relate this bathymetry data to biological data that we've been collecting over the past few years. To give you an idea of what you're looking at, the colored portion of this image starts at the top of the wall. The orange and red colors are basically at around 40 meters deep, right as the wall drops off if you've been diving out there. And then as it goes into the yellow, we're getting down a few hundred meters almost instantly. And then into the green color, you're down close to 800 to 1,000 to over 1,000 meters deep, very, very quickly in close proximity to the shoreline. We have a number of ongoing projects I just want to touch on today. We're starting to investigate marine mammal abundance and diversity with the help of Charlotte and Diane in Abaco. We're looking at sargassum communities, impacts of fish aggregation devices, pelagic shark ecology and conservation, and broadly trophic ecology. So I'll tell you a little bit about each of these in the rest of the talk. So marine mammal diversity and abundance, you know, we're out there on the open ocean all the time. And so we started to notice these marine mammal species that we just hadn't encountered before, and it got us really interested. And so we really just wanted to contribute to a lot of the ongoing work that's been taking place in Abaco and elsewhere. So we're really just collecting baseline data on what's here and trying to contribute to existing data sets at this stage, hoping to build on that in the future. Our current research questions are basically what species are here, how abundant are they? And that's where we're starting for now. We answer those questions with visual transects, basically boat-based surveys. And while we do those, we're not only observing marine mammals, but also seabirds and pelagic fishes. We've put out one hydrophone thus far so that we get better temporal coverage of presence and absence of marine mammals. And we hope to add a fixed-wing drone next year to conduct aerial surveys as well. So those visual transect surveys take place just off of Cape Eleuthera. You can see two of them marked here. One is quite close to the drop-off and another is a couple kilometers off. They're about 10 to 12 kilometers long each, and we run them weekly. Well, we did until coronavirus anyways. So since 1994, there have been 13 species of marine mammals documented in this northeast corner of EXUMA Sound, ranging from humpbacks that are incredibly rare, but a real treat when you find them in 30 feet of water near, in our case, near Deals Point, the one time it happened, to pilot whales and sperm whales that seem to move through just on occasion, to beaked whales that are more resident. So there have been about 100 sightings since 1994. Most of those come from data that Diane and Charlotte have been keeping, and we've been adding sightings from our visual surveys from 2018 onwards. And we've really enjoyed this work thus far. Also, every time we see marine mammals, we take photos, and hopefully we can someday contribute these to ongoing data sets as well and get an idea of residency, returns, migrations of these animals. These pilot whales, for instance, could be coming from the east coast of the United States. So here you can see, this is a figure from the undergraduate thesis that Alexa Hoffman did just earlier this year. And you can see some of these sightings overlaid on top of the map of Cape Eleuthera. Each of the points represents a number of individuals. The color dictates number of individuals. These aren't just marine mammals. These are also seabirds and pelagic fishes, flying fish, all kinds of things. And you can see how they roughly line up with each of the two transects that we do. And so what we hope to do is relate these sightings to environmental variables, look at seasonality, when these different species come through, look at the bathymetry of the area where they seem to be congregating, see if there are any features that lead them to feed in one place. We've seen some really cool feeding interactions between different species. So we hope that we get some really good data out of this, but we need to do it for a few years before we really dig in. We've also been investigating the sargassum communities. Sargassum is an essential fish habitat. It's the only physical structure in much of the open ocean. It provides refuge habitat for larval fish and invertebrates. And so all kinds of pelagic predators feed in these communities. So naturally, we were interested in seeing how they fit into the wider food web of the epipelagic zone and how much do pelagic predators rely on the sargassum. And that's where we started. It quickly got more interesting. We realized that the sargassum that we find offshore is not always one of the two species of true pelagic sargassum, which you can see to the left of your screen. Instead, some portion of it was coming from right up along the shoreline where benthic sargassum gets ripped up by wave action and then drifts out to sea. And you can see that on the right. So building off those observations, we decided we first needed to characterize the communities living in various sargassum species and then investigate how the sargassum species connect coastal and offshore ecosystems. We have a long way to go in answering those questions, but we've started to get a sense of how much of the sargassum off the coast is actually the pelagic variety, like you can see here. And it turns out that it's really not very much. A lot of it is the benthic variety that drifts out to sea. But the communities do seem to differ. In the pelagic species, we find organisms like sargassum nudibranchs, pipefish like this one, some really cool unexpected critters like this pycnogonid or sea spider. And then in the benthic community, we see a different assemblage. And not only is there sargassum drifting out there, but with it, all these other coastal varieties of algae and even casuarina needles. Sometimes we see whole weed lines made up entirely of casuarina needles out in blue water. And so the species we find in those coastal varieties include more hermit crabs and sea slugs and snails and mollusks and these kinds of things. So we expect these species to be carried out to the open ocean from the coast, where they can then provide an additional source of food to pelagic predators. And at the same time, those pelagic species are getting carried in to the coast from offshore. So there's a lot of exchange happening. So our next steps in this line of research are to first develop a really comprehensive species ID guide, because right now it takes so long to just figure out what we're looking at. And then estimate the percentage of blue water sargassum that comes from the coast or is one of those two pelagic species. Estimate the biomass of each and relate that to environmental variables, and eventually quantify how much of it ends up in the deep sea. You know, on a bunch of submersible dives over the past few years, we've seen a lot of sargassum down on the bottom. So it's not only connecting the coast to the epipelagic zone, it's also going from the coast out to the open ocean and then sinking down to the deep sea. I'll just touch very briefly on fish aggregation devices. We've been doing a lot of work on them, call them FADs. I know Eric presented on some of this earlier in the webinar series, so I just recommend that you check that out if you get a chance. The two major questions we've been focusing on are how does the diversity of a FAD-associated assemblage change over time? And then how can we track fish in 3 dimensions around a moored or a floating FAD? And we're studying this primarily because there's a lot of bycatch associated with fish aggregation devices in purse seine fisheries. And so we're trying to come up with information that can inform bycatch reduction in those fisheries where they take place. And then pelagic shark ecology and conservation. We've been doing a bunch of work, again, just starting with what species actually exist in EXUMA Sound. And we've been running pelagic longline surveys. I won't get into those results, but we found there really aren't a lot of pelagic sharks out there off of our coast anyways. We've been investigating silky shark movements across ontogeny and also investigating silky shark feeding ecology. The silky shark is listed as vulnerable. It's most commonly captured as a non-target species in FAD fisheries, purse seine fisheries, and it makes up about 90% of elasmobranch bycatch in those fisheries, and mortality can be really, really high, over 90% when they're caught. It's also an important species in the fin trade, falls out near the top of ecological risk assessments for pelagic sharks, and it's experienced dramatic declines somewhere between 50 to 90+% over the past 3 generations. So a lot of folks are interested in getting more data on silky sharks to reduce bycatch interactions and hopefully support the recovery of silky shark populations in the Western Atlantic. We need to collect a lot of data on their critical habitats, figure out where they spend their time at different points in their lives, and look at how much mixing there is between different populations throughout the region. So we do that by deploying satellite tags that collect temperature and light and pressure data, so we can look at what temperatures they prefer, where they're going, and what depths they hang out at. So far, we've deployed over 25 tags from anywhere from 8 to 242 days at liberty on sharks that are anywhere from 96 centimeters, we're talking like 1 to 2 years old, to very large mature adults that are around 250 centimeters long. We've deployed tags in the US, the Bahamas, Haiti, and Belize. And you can see some of those deployment and pop-off locations here scattered around the eastern US and a little bit in the Gulf. We've added a bunch in Belize recently that aren't on here, and some in the Bahamas as well. So zooming in, you can see with the help of a lot of different folks, we've put out tags in Tongue of the Ocean, EXUMA Sound, San Salvador, and Mayaguana as well. It's been really cool thus far. A lot of the animals we've tagged in the Bahamas have actually stayed here and haven't traveled that far. Animals in Tongue of the Ocean seem to stick around there. The animals that we tag near Eleuthera or San Salvador seem to stick around this area. And so we're hoping to build on this data set over the next 1 to 2 years and really get a good idea of where these animals are moving. Also, I mentioned we can look at their dive behavior and their thermal preferences. You can see here, these are the temperature and depth data for one shark. You can see depth from 0 to 600 meters on the y-axis, and then the date on the x-axis. And you can see for the vast majority of the time this animal was tagged, it was spending pretty much all of its time in the top 200 meters, occasionally making deeper dives past 300 meters. And then at some point around January 9th, 2020, if you can see there, it starts to exhibit a very different dive profile. And that's probably because it got eaten right around that point. We're also looking at their feeding ecology through gut content data, stable isotope analysis, and fecal swabs as well. Try to get an idea of how they fit into the wider pelagic food web. And that ties into our tropha ecology theme. Our big research questions here are to figure out how these consumers connect ecosystems across ontogeny and ultimately develop ecosystem models of EXUMA Sound. So if we can do that, if we can predict how these ecosystems will respond to stressors, it'd be really great because we can manage them a lot more effectively. Currently, only 2% of important bathymetric features, which technically include steep walls, seamounts, ridges, and canyons, actually fall under a designated protected area. And the main reason for that is simply because we haven't studied them enough. So we have a lot of work to do, and we've really just gotten started. So our team is really massive. I just want to say thank you to all of our students at DCMS and the Island School, visiting groups, of course, our research team at CEI, most importantly, and then all of the folks who have been supporting us and working with us over the past couple of years. And lastly, I just want to say thank you to BNT for hosting the conference and for their support of our research over the years.
Thank you, Brendan Talwar, for sharing your research project on the ecosystems in the Bahamas. I will invite our final presenter for today, Brendan Shea from Beneath the Waves, to share his screen and to speak to us on deep water habitat use of tiger sharks in the Bahamas.
Okay, thank you. Can you hear me now?
Yes.
Okay, great. Well, thank you so much. We're so happy to be here today talking a little bit about our research. I'd first really like to thank the Bahamas National Trust for putting on this conference during these unprecedented times. Also the Department of Marine Resources in the Bahamas, Dr. Lester Gittins and his team for their permitting support over the last couple of years. And all of my co-authors on this presentation. It's a rather lengthy list over there, including researchers from Beneath the Waves, Trinity College in Dublin, and University of Miami. We just want to thank all of them for their contributions to this project so far and going forward. Oh. So if you've been watching these webinars, as Steve kind of touched on, we are not the only group who is studying sharks in the Bahamas. The reason for that is the Bahamas is one of the best places to study sharks in the world due to the protections that are afforded to these animals. The entire exclusive economic zone was designated a shark sanctuary in 2011, but really the protection goes back further than that, back to 1993 when the longlines were banned. Even in otherwise protected areas, longlines can be a potentially significant source of mortality for sharks. So between that longline ban and the subsequent designation as a shark sanctuary, sharks are really protected in the Bahamas in a way that they are in a few other places, if any, in the entire world. Now, this has been good for sharks. It's also been good for the Bahamas. Estimates put the economic contribution of shark tourism to the Bahamas economy at over $100 million a year through shark diving and things like that, which makes the Bahamas a great model for future large-scale MPAs designed to protect sharks. But to kind of get there and really accomplish that, we really need to have a better understanding of how are sharks using the sanctuary, what in the sanctuary is benefiting these sharks the most. Part of that, and the reason we're all here today, is one of the unique features of the Bahamas is these geographically nested deep water features. Brendan had a great shot of the wall in that last presentation. In these features, we have incredible bathymetric gradients where it goes from 100 feet to thousands of feet and across a really, really short distance. So as shark researchers, we're really interested in knowing how are sharks using this deep water. We know, as Steve touched on, there are some sharks that really specialize in deep water, but increasingly from a lot of tagging studies, we're realizing that many of what we would consider to be those shallower sharks, blue sharks, mako sharks, tiger sharks, as well as lots of other elasmobranchs and teleos species, are making a lot more frequent use of deep water than we thought. Whether that's diel migrations, where they spend the daytime at deeper, cooler waters and move up to the surface at night to feed, or animals that are diving down to feed on the biomass that may be down at depth, like Charlotte talked about with the whales during her presentation. In particular with tiger sharks, we do know that despite being service-oriented sharks, they are capable of pretty incredible dives. Dives to over 1,000 meters have been documented off of Brazil and off of Australia. But there's this really high degree of intraspecific variation amongst tiger sharks. So regionally, we're really interested in trying to understand what kind of role do these deep sea features play in the life history of tiger sharks. Quickly, the reason we're so interested in tiger sharks within the shark sanctuary is tiger sharks are really the apex predator of the shark species in the Bahamas. And unlike things like Caribbean reef sharks, which are a little more resident, tiger sharks are highly migratory. So while the shark sanctuary is affording them protections for parts of the year, they also might be spending huge parts of the year outside of the sanctuary. On the right, you'll see an animation playing. That's a shark that a tiger shark we tagged in May 2019. And that's a one-year track. During that time, it swam all the way up to off the coast of New England before heading back down to the Bahamas. So huge portions of the year are spent outside the shark sanctuary. So it's increasingly important to understand what the benefits that the shark sanctuary are conveying onto the sharks are, given how they might not be within that protected area for their entire year. So our research questions around this topic were, how are tiger sharks in the Bahamas using the available deep water habitat? Kind of going beyond that, what are some of the potential drivers of vertical habitat use by tiger sharks regionally? Our study locations for this project are two areas we've been working in pretty extensively for a few years now. That's Nassau, New Providence area, and that includes some of the islands extending up to the northeast towards Eleuthera, like Rose Island in this chain here. We do a lot of work out there. We also do a lot of work down in Great EXUMA, and these are actually both perfectly situated to look at this deep sea question because they're located right next to some of those deep sea features that have been pointed out in previous presentations, the tongue of the ocean here, EXUMA Sound here. So to get at these questions, we started doing some tagging. Most of the work that I'm going to discuss today was completed in April and May of 2019, again in those areas around Rose Island, Nassau, and Great EXUMA. We used a total of 7 pop-off satellite archival tags, or PSATs, which are the tag that Brennan just demonstrated in his last presentation. It records temperature, depth, and light levels to give us an approximate idea of location. We also had 6 deployments with biologgers, which are these customizable multi-sensor packages. These ones we used actually had integrated cameras to allow us to get point of view footage from the perspective of the shark. It also measures water temperature, water depth, internal temperature of the animal, and movement in 3 directions. It's a triaxial accelerometer on there that allows us to measure acceleration in 3 directions. Similar to a pop-off tag, this whole thing does release, and we have to go collect it from the surface of the water. As you can see it floating there in the upper right panel. Moving to some of our results quickly, this series of figures is similar to the one that Brendan just showed, in that it's a time series showing the depth preferences of different tiger sharks. You'll only see 5 on this screen. That is because we used different deployment durations for the 7 tags that we deployed. Those deployments were for up to a year. So on the deployments that were longer than 53 days, we actually had them sampling only depth and not temperature in order to maintain better memory while still sampling at a high rate. So you can see those depth profiles here. On the left side, that's a tiger shark that was a 4-month duration, and that shark swam up to northern Florida area from Nassau. By the time it popped off, you can see there's pretty consistent diving to below 200 meters. And this shark here on the right, that is actually the depth profile from the shark that swam up to Cape Cod. So that is a 1-year track. And keep in mind that the shark is not within the Bahamas for much of this track. It is up in other parts of the ocean. But we still see these really strong vertical habitat preferences with some regularity. You see times where it tends to be more surface oriented, but they're definitely spending a lot of time down in deeper water. Focusing in on the Bahamas, and just with the period of time that those sharks that left the Bahamas were only the time that they're within the exclusive economic zone, we found some pretty striking things about how the tiger sharks are using the deep water here. All the sharks we tagged were found to dive to 200 meters or more at least weekly. As you can see, the patterns of how frequently they dove really varies tremendously among sharks. This shark here in the upper left spent a lot of time diving and a lot of time just in deeper water, as compared to the shark in the center here, which had very sporadic dives. But all of them dove to at least 200 meters, at least weekly. And most of them, honestly, at least had periods where they were diving closer to every day or two to depths of 500 or 600 meters, which is pretty amazing. Put together as a set, the sharks in the Bahamas spent 7.5% of daylight hours at depths of 200 meters or more, so almost 10% of their time. And there was some variation in that. Some sharks spend as much as 20% of their time during the day or more down in the mesopelagic zone at 200 meters or more. So they're really spending a lot of time down at depth. We did not notice any potentially dramatic patterns in their day-night distribution. This is just a series of histograms showing their depth preferences by day and night. Day is on the left, night is on the right. As you go down each histogram, that indicates a deeper depth bin. So again, we just do see tremendous variation amongst sharks. Some sharks, like the one in the upper left, spending almost all of their time distributed evenly throughout that water column, whereas other ones are extremely surface oriented. When we started to incorporate the camera tag footage and accelerometer data is where things really got interesting. This is a typical point of view shot that we would have of a tiger shark swimming here. And I'm just going to kind of be skipping ahead through this video as it makes a dive just to demonstrate a couple of things. As you can see, the shark is just swimming. This is what we consider pretty standard swimming behavior. You can see its head moving left and right, potentially just searching for some food as it moves along the seafloor here at about 300 feet. But as it skips forward here, and the shark's going to start to dive, what gets really interesting is you'll notice the shark's head is not moving nearly as much. And what that indicates to us, combined with that accelerometer data that I'm not going to go too deep into here today, is that the shark is really gliding. At this point, it's no longer actively swimming so much as it's kind of just gliding down the wall. And as it gets to deeper and deeper water, this trend kind of continues. 600 feet down to 1,000 feet. It's eventually going to move down to 1,200 feet and then into complete blackness at 1,300 feet or more. The little red dots you see flying through the water there are just little bits of particulate that are being illuminated by the laser on the camera. But it was pretty striking to us to see how little these sharks were actually swimming on their way down. So this camera footage, kind of coupled with some of the accelerometer data that we were able to look at, we're still processing some of it, it's not completely worked up yet, gave us some insight into a couple of potential drivers for this diving behavior, and kind of some perspectives on other drivers that have already been proposed. One is thermoregulation. There's kind of two reasons why animals might be diving to thermoregulate. They could actually be diving to cool down if they're in warm surface waters. They might be diving down to deeper waters to lower their core temperature. Or if animals are diving to feed, for example, many of them might have to return to the surface to actually warm back up. In the case of the tiger sharks, the evidence we have so far would support the latter, that they are diving out of their thermal comfort zone, either to feed or for some other reason, and then returning to the surface because they do need to spend some time at the surface to warm back up before they're able to perform any additional dives. There's also potentially a social or reproductive component to this diving behavior. Our accelerometer data revealed on one large female tiger shark down at 500 or 600 feet, it was contacted very hard. Either that or it potentially swam into something, but I don't know how likely it is that a shark would just swim into something when it can still see where it is. And this kind of hard bump, particularly on a large female, definitely kind of raised the question to us, could there potentially be a reproductive role for the deep sea? Is that a male shark attempting to make a mating attempt on this large, reproductively mature female? And the third one, which I think was potentially well demonstrated in the last video that I just showed you, would be energy efficient foraging. Tiger sharks are known to display oscillatory diving patterns in a lot of areas throughout shallow seas as a means of kind of conserving energy while they're searching for prey. The Bahamas, with this unique wall, very plausibly could serve as a way for tiger sharks to perform extended foraging searches, looking for benthic prey along this huge bathymetric gradient while exerting little to no energy as it just kind of glides down the wall. This has some very interesting implications going forward that we are continuing to work on with some ongoing research. Steve touched briefly on some of the work we're trying to do. We're expanding our acoustic telemetry arrays to include deep water receivers off EXUMA and NASA. Those are already down, so the next time that we download those receivers, we should have some interesting information on how sharks might be using the deep water. We are also increasingly using deep water brubs, or baited remote underwater videos, dropped down to hundreds of meters or more. Hopefully we can use those to capture feeding or other behaviors down there to shed more light on what the role of the deep sea might be for tiger sharks. The implications, I think, are very important when we talk about long-term protections for tiger sharks and MPA design. It certainly seems to us, based on this research we've done so far, that the deep sea is playing a largely uncharacterized and perhaps underappreciated role in the life history of tiger sharks, at least regionally. So if we're going to be citing more MPAs to protect large sharks like tiger sharks, I think we really need to kind of disentangle the drivers for why sharks are driving to be able to cite those MPAs correctly and make sure that we're affording these animals the most protection we can. I also think it sheds really interesting light on potential connectivity and the role of tiger sharks as a potential keystone species. Brendan just talked a little bit about how connected the shallow and deep sea ecosystems truly are. And based on how frequently we're seeing these large animals, large apex predators diving throughout the water column, they're certainly redistributing nutrients and organic matter throughout the water column while they do that. So there's plenty more research to go. We're just getting started in this long project of studying the deep sea and how sharks are using it. But what we are finding so far is that this kind of last frontier might play a much bigger role than we previously realized. With that, I'd really like to thank just again the Bahamas National Trust for putting on this conference, as well as a couple of our partners down in the Bahamas, the Grand Isle Resort and Stewart Coves, who have been absolutely critical in our success down there over the years. And thank you very much.
Thank you, Brendan Shea, for sharing your findings on tiger sharks and sharks in general, their behavior and connection with the deep sea. We would like to thank you guys for sharing your data for today. We hope that you can join us for the remaining sessions scheduled to continue every Wednesday into August, which is now. As always, we welcome your questions, feedback, and support as the BNT team continues to pivot our programming towards new normal methodologies, including changing the platform of our conference to virtual reality. There is still a need for funding events like these weekly sessions. We would be grateful if you consider donating towards the success of the Bahamas National History Conference 2020. If you are enjoying the Bahamas National History Conference virtual webinar series, we simply ask that you consider giving a donation in any amount as a means of showing your support. An email with a link for donation will be sent following this session. So I'm going to open the floor for questioning and answering. We have some questions that were answered already by the panelists in the question and answering. Okay, so we have questions from Facebook for Dr. Charlotte Dunn. How were scientists able to determine blue whales could communicate across the Atlantic?
So a long time ago, a pretty famous acoustician, Dr. Chris Clark at Cornell University, was famously given access to Navy hydrophones, and they have hydrophones all over the planet, basically. And he was able to use the hydrophones to determine that information.
Thank you, Dr. Dunn. Satellite tag and telemetry work was fascinating. Why did the shark camera need a laser? Just wondering what the laser did. I think this is for Brendan Shea.
Yeah, sure. That's just to help illuminate things in front of us. We don't use like bright white light because that is a little more disruptive to animals down in the deep sea, whereas that red laser does not affect the behavior of animals nearly as much.
All right, thank you for that response. And this is for Brendan Shea again. It's not quite the deep sea, but deep in Sapphire Blue Hole lives the Bahama cavefish. Is it protected, endangered, rare? Anyone study it? Do you know of that?
I cannot speak to that personally. I'm not sure if any of the other panelists might be able to, but.
Okay, thank you so much. If you have any more questions for the panelists, please submit them in the question answering section as well as comments on Facebook. Another question, any signs of swordfish? Curious. This is from Facebook Live.
This is Brendan Tallwar.
Yes.
We haven't caught any on any of our pelagic lines. I'm sure there's some around, but we haven't caught any or seen them.
Thank you. And a question for Dr. Dunn. What can persons do in addition to educating others to help bring awareness and protect marine mammals in the Bahamas?
Well, as Frankie knows, another one of our interns, we take Bahamian interns and we also run whale camps to try and disseminate that information. We've got an education package that we're trying to get into the schools. But yeah, one of our biggest problems here is that people don't know we have whales, and something that ironically the manatees, because they're coming into shore, and although we don't recommend touching them or giving them water, people are actually able to see them. And the manatees are really helping Bahamians understand that we have marine mammals more than just dolphins. But it's hard to see whales in the Bahamas because, most people don't get into a boat that goes into the deep water. And a lot of the whales aren't at the surface all the time. But yeah, go to our website. We post regularly on social media, and apply to come as an intern, and then tell all your family what cool animals you saw. I think when Frankie was here, we had some great encounters with dolphins and sperm whales. So yeah.
Thank you, Dr. Dunn. What is the accepted mortality rate when you set the long lines? How many sharks don't make it?
This is Steve Kessel. The ideal mortality rate is 0, and that's what we're aiming for. With sharks, they're not impacted as much as teleos when you bring them up from depth from a swim bladder perspective, because they don't have one. So they're able to transverse the depths even, at a greater speed, better than most fish species. The question, probably the biggest factor is going to be temperature, the temperature difference between the depth we're bringing them from and to. We bring them up pretty steadily to help them acclimatize to this. But really, when you let them go, you're making an assumption that they survive. I don't know to what extent these species have been studied in terms of post-release mortality. I'm not sure if, Brendan, if you guys have done any of that with sat tags on some of these species. I know some of the bigger species like the blunt-nosed six-gill in the Gulf with Dr. Grubbs and Brian, they've done some of that work and seen that they do survive. So we're hoping that, like a lot of these other species, these sharks are pretty tough and pretty hardy, and we do everything we can to minimize the impact. So the answer is the ideal mortality rate is 0. I don't know if we have really set a bound for acceptable, but I would expect that most of the sharks I caught on that trip would have survived.
Thank you, Dr. Kessel. Another question: how does testing of nuclear and other weapons in the remote oceans affect our marine mammals? Dr. Dunn?
I don't know about nuclear testing, but it's the same with any if there's any loud sound, then they will be subjected to the impacts I discussed. If there's explosion type sounds, I don't know if that's how they get rid, for instance, of nuclear waste, but then animals that stranded here in 2000 that were obviously very close to the Navy ships that were putting out the sound, when we did an MRI on those animals, their inner ear cavities had actually burst. So if they're very close to the sound, not only will they strand themselves to get away, but they will have suffered physical damage from that sound as well.
Thank you, Dr. Dunn. That seems to be all of our questions from Facebook Live, as well as here in the question answering section. This session is being recorded and is streaming Facebook Live. After today's session, the recording will be available to all by visiting the Bahamas National Trust Facebook page and YouTube channel. If you'd like to keep up or get in touch with the Bahamas National History Conference, do so by visiting our website, BNHC242.com, and Facebook.com//the BNHC to follow social media updates. Questions and suggestions can be directed by email to bnhc@bnt.bs. We want to thank all of our panelists for a great and interesting session today. And thank you all for the questions for our panelists.